Good morning, D-Wolf biology students. It's me, Mr. Cheney, with another brilliant tutorial. This one is from the textbook. It's for the Interactivity Osmosis lab in the textbook, the SAVAS textbook. So in Google Classroom, you will find it here, titled Interactivity Osmosis. So we'll click into it and let's get started on that as it's loading up the material. So osmosis is the transport of things inside and outside of a cell. So it's kind of a different process when we're talking about plant cells versus animal cells. And so we'll kind of talk about some of that. Okay, so here we go. We have 6 slides to go through on this interactivity. Osmosis is the diffusion of water through a selectively permeable membrane. Permeable means that things can get through it. Water molecules pass through cell membranes using embedded protein molecules called aquaporins. Since water does not flow directly through the cell membrane, osmosis in cells is facilitated diffusion. As with diffusion, water moves from areas of high concentration to areas of low concentration. When the concentration is the same on both sides of the cell membrane, the system is at equilibrium. So, as always with textbook stuff, when it tells you to do something, you need to do it because it's a self-grading thing. So on this particular slide, it wants us to see the difference between the outside of the cell and the inside of the cell, and how things are transported. So it's showing water going through the aquaporin right there. So we're good to go there. We can go to slide 2. All right. The instructions for slide 2. Osmosis. Look at the concentration of sugar molecules on either side of the selectively permeable membrane. Sugar molecules cannot pass through this membrane, but water can. Draw lines on each side of the tube to predict the water level once equilibrium is reached. Okay, so you're going to be using your drawing tool to figure out. Okay, so that's the selectively permeable membrane right there. Okay, and then we got sugar molecules. There's a high concentration of them over here. Your water level is here, and so what they're asking you to do is draw lines where you're going to predict on each side of the tube where the water level will be once the equilibrium is reached. So at this point, there's way more sugar molecules over here than there is over here. So what do you think the water levels will look like when there are equal sugar molecules on each side? So when you come over here, you do your draw tool, pick a color, and then you're just going to make your guess. So you might think that the water level on this side will be here, and then over here it might be here, but this is going to be a guess on your part. So that's what you want to do there. Slide 3. The diagram compares diffusion of sugar molecules and osmosis. Explain the difference between osmosis and diffusion in cells. So it's explaining diffusion right here with the lipid bilayer, and then it's got osmosis down here. So you can see one of the big differences is that with osmosis, we have this transport channel that we've talked about versus no channel over here. So that's one major difference. Okay, and there are others that you can talk about in slide 3. Number 4, we'll click on introduction. It says living cells are surrounded by liquid. Osmosis and other types of diffusion occur constantly to maintain equilibrium. A change in the concentration of solutes outside of cells can have immediate and drastic effects on the cell. Click on the hotspots to learn about these effects. So this is one of those slides where you want to click on each one of these gray circles. All right, so this is isotonic. This kind of solution has the same exact concentration of solutes as the cell. Water will move in and out of the cell, but at equal rates. The cell will maintain its normal shape in this environment. Okay, we'll click on the second gray circle. This is hypertonic. Cells placed into solutions with higher concentrations of solute will experience a net movement of water out of the cell. This movement of water will cause the cell to shrink. And lastly, we have hypotonic. A cell placed into an environment where the solute concentration is lower will experience a net movement of water into the cell. So this movement will cause the cell to swell and may eventually burst. Okay, so hypotonic means water is coming into the cell. Hypertonic means that it's going out of the cell, so it would shrink, and then isotonic is more of an equilibrium thing. All right, slide 5 out of 6. Okay, the instructions for this one are: cells are placed in beakers containing different concentrations of solutions. For each tile, compare the concentration of particles inside and outside the cell. Drag the tiles to the appropriate box to describe what will happen to the cell. So it'll tell you if you're right or wrong. So on that particular one, cell does not change because there's an equilibrium of particles out there. On this, we see that there's a smaller amount of cells inside of the membrane and lots of cells on the outside. So does that mean that it's going to shrink or swell? So you're going to match those to the 3. And then we have last, slide 6. So now let's look at an example in a plant cell from a leaf. Use the slider to examine a plant cell before and after the plant was watered with very salty water. So after watering with salt water, the plant wilted. Propose an explanation as to why the plant wilted using evidence from the cell diagram. Okay, so this is before the salt water, this is the after salt water. So you can see that the central vacuole, okay, the storage water of the cell, okay, shrunk considerably when you actually use salt water. That's because salt actually draws water out of things. So you want to answer based on that diagram right there. Okay, and that's that. That is the interactivity osmosis. I will attach this to the assignment in Google Classroom as a tutorial. And I'll see you back in class.